Well, it's great to be here and great to see so many of you here. As Bob mentioned, I'm Ewan Ashley. I'm an Associate Professor of Cardiology and Genetics and Biomedical Data Science in the School of Medicine. And it's really my pleasure to be here at the Big Data and Biomedicine Conference to introduce a really interesting session. In particular, a pleasure because these are not just my colleagues, but my friends. And I would say that the title of this is Cardiovascular Big Data. And cardiovascular scientists have really been interested in large-scale data for many, many years. If you think back to the population health studies, like the 7-country studies in the 1950s, I think cardiologists and cardiovascular biologists were interested in large-scale data to try and tell us more about health and more about disease. Of course, the very famous Framingham Heart Study in Framingham, Massachusetts has been going over multiple generations. And I think what you're going to hear in this session is really the bringing up to date of those kinds of studies. We're certainly very interested in technology. It's not going to be news to many of you that as cardiologists we're very into gadgets and we're basically geeks. So this is a world where we have gadgets that can give us a window onto the cardiovascular system. Some of you know that here at Stanford, we launched a few years ago in collaboration with Apple, the My Heart Counts study, and that was one of the fastest recruiting studies of all time, partly because it was announced by Apple, and people pay attention when they do things. We had 10,000 people sign up in the first week. We had 30,000 people sign up in the first month, and the study is now somewhere between 50 and 60,000 people. We're on to the second generation of that study, which is now one of the first randomized mobile studies. So if you're interested in coaching approaches to increasing physical activity, or you're just interested in your health, or you'd like to participate in the study, then I would invite you to do that. It's a free download from the App Store. And then even today, we were just releasing some new data looking at just how accurate the wearable devices that many of us have on our wrists actually are. Increasingly, our patients are bringing us data from these devices. And so we're very interested to know if that data is reliable and if we can in good faith use it for the management of our patients. Well, that's enough from me. It's really my job here is to introduce the talent, and I'm very happy to do that. The first speaker of the session has an accent not too dissimilar from mine. Actually, he comes from my native land, Scotland. He comes from a small island just off the west coast of Scotland called Skye. So happens they make a great single malt scotch there. It's called Talisker. It has a very smoky... Am I supposed to be introducing him? Oh, yeah, sorry. So Calum MacRae is Chief of the Division of Cardiology at Brigham and Women's Hospital. He's a Professor of Medicine at Harvard Medical School and runs the Cardiology Fellowship Program there, as well as playing a lead role in the cardiovascular genetics clinics. He's really been a pioneer in very many ways, laying out the early data for the foundation of cardiovascular genetics diseases like hypertrophic cardiomyopathy and other inherited cardiomyopathies. And in the most recent era, and he'll tell you a little bit about this morning, he has been leading one of the new ideas, which is the One Brave Idea from the American Heart Association. So I will leave it to him to tell you more about that. Please welcome Calum MacRae.
I'd like to obviously thank you for that introduction, and also Dean Miner and the other organizers for the opportunity to speak. As Ewan pointed out, my accent does share some similarities to his. I have a challenging task, which is to describe a project that really has only just begun to get started. But I think, having heard the earlier talks, will actually put some of what you've heard already in some perspective. Our project, named the One Brave Idea by the AHA, the actual idea was really defined here as redefining coronary heart disease at the transition from wellness. So I'm going to really try and explain to you how we came about that strategy. I think one of the things that is quite clear when you listen to people talk about precision medicine or about the introduction of data and AI into medicine is that there are sometimes some competing visions. These two slides were taken off the web on the same day. At one point, large pharmaceutical companies think that they're going to use very precise, usually single discriminants to identify subsets of disease, while technology and informatics are being introduced into healthcare really around business models that are ancient and somewhat broken. And the reality is that both of these approaches are actually failing in the current time. And they're failing not really because of any intrinsic abnormalities or problems with the strategy, just because of the implementation approaches that have been used. I actually think the other thing that's salutary in this lesson is, and you've heard some of it outlined already in a couple of talks, Eric mentioned it, I think, quite clearly, Josh also highlighted it, is the fact that at the core of most of the problems in understanding how we're going to be able to do this in medicine is really a fundamental defect in information content. And it's actually quite interesting that in almost every sphere of medicine, and this is probably why this will actually be the era in which precision medicine and AI finally reach medicine, simply because of the fact that now there's a convergence of all of the problems around this one theme, is that there's a core deficit in the phenotypes that we're actually measuring. So you can see on the left, a geneticist's vision is that you can study very large effect alleles in that top left-hand corner, very small alleles in that bottom right-hand corner. No geneticist worth his salt fails to have a diagram like this in their talk. But the reality is that the majority of genes or genetic effects are actually sitting in the middle. They're major effects that are only present in the presence of a conditioning variable which we never measure. And as a result, you can't even tell that they're truly heritable. And many of the assays that we use in medicine are essentially legacy digitizations of phenotypes that were probably present in the Middle Ages, but certainly present 200 years ago when Osler was around. The same defect in phenotyping has become very transparent in genomic medicine. It's almost impossible to take the 5 or 6 variants that you're left with and understand mechanistically which of these might cause the disease if you don't know anything about the biology at a cellular level, at a molecular level, that the actual patient in front of you is suffering from. Precision obviously suffers from exactly the same lack of dimensionality. And one of the most, I think, dispiriting elements of the digitization of healthcare is the failure of multiple trials of monitoring the same stuff we monitor in hospital, but monitoring it at home, to have any impact at all on outcomes, even relatively modest outcomes like heart failure readmission rates in the modern era. And so ultimately, phenotype is rate limiting for a variety of reasons. We take static and cross-sectional views. Almost none of them are standardized. There are essentially 2 standardized traits in all of biomedicine, the international normalized ratio and the 12-lead electrocardiogram, both of which cardiologists should be proud. But I challenge anybody to actually find any other global standard. Almost every CT scan that is done, almost every MR that is executed on, is using a completely arbitrary protocol, usually defined by the person who's running the machine. And so as a result of that, we're really stuck with things that are half-baked efforts to try and make the best of data that we've been collecting over the last couple of 100 years. And I won't even talk about conditioning variables, since we really only measure 2 of those, alcohol and tobacco, and that's probably an almost infinite data set. So this is really, I think, the most obvious gap. We started in this project thinking about coronary disease, and the phenotypes that are measured in this, the most common cause still of death in the developed world, are really a sequence of events that are actually almost embarrassing to relate. We measure LDL because you can see it with the naked eye when you sit samples from very premature coronary patients on the bench. The electrocardiogram, it was almost 15 years after the first myocardial infarctions were actually documented that people realized that there was an electrocardiographic feature. Almost every other part of the disease that we look at is late, and usually nondescript. It's a final common pathway in a whole slew of disorders. And one of the things that I think is becoming evident is the only thing that's truly common about common disease is the superficiality of the phenotypes that we use. The electronic health record, I think, is a great example of that. I'm not sure I even care what I thought last week about a patient if it requires natural language processing to extract it. And so the saddest thing, I believe, is the fact that there's this remarkable sense that we have these perfect phenotypes if only we could measure them, but we don't really measure anything very effectively. And in fact, genomics is probably the most important advance in all of biomedicine in the last several hundred years for 2 reasons. 1, it actually is standardized, and 2, it's a bounded data set. It's the only bounded data set, I believe, in all of biomedicine. When Ewan and I were participants in the undiagnosed, we still are participants in the undiagnosed diseases clinic, we saw that when you add up all the phenotypes in biomedicine, they reach the grand total of about 9,800 phenotypes. So it's hardly surprising that we're unable to deconvolute genomes, proteomes, connectomes, etc., that are orders of magnitude more complex. And I certainly wouldn't hesitate to be negative about the exposome, which we've really failed to measure completely. I think the idea that we had coming into this project was that we needed to really begin to build some formalized metadata that would allow us to anchor all of these planes of data across diverse data sets, from a single dipole moment in the membrane all the way up to a population. And there are really only two things that allow you to do that, I believe, and those are small molecule and biophysical challenges, perturbations. And it's worth pointing out that even the relatively modest and fairly straightforward geospatial anchors that people use in many other vertically integrated data sets simply do not exist in biomedicine. The other thing that I think this project was designed to do is to really think through how we move from broad to deep and do it in a strategic manner by design, rather than thinking just about what we've previously done and why we've done it. The idea was referred to earlier as the one brave idea. I think the bravest thing about it was that the initial application required only 250 words. And so I think that was certainly the best ratio of words to revenue that I've achieved in my short life. The other thing that I think is quite interesting about it is it's being run, and I think very effectively run, in a very different way for a research program. There's a cohort of investigators that work on a very goal-directed fashion, report up through the AHA, but then have the funders, the American Heart Association, Verily, and AstraZeneca, all really participating in a board-like structure to follow the trajectory. There's a core team that was involved, includes Ewan, several other folks that you'll recognize if you know cardiovascular or information science. But the bottom line is a diverse group of individuals going all the way from single molecules through to population, and including some venture capitalists and others with experiences that are considerably more diverse. We recently hired Stan Shaw as the Chief Scientific Officer. He was a participant in the research kit development for diabetes. The core element that we thought we would focus on is that existing risk factors really reflect very late stages of the disease process. Most of the abnormalities that are detectable in coronary disease are not detectable until there's actually a terminal or a endpoint event. There are some biomarkers that are present, but even those biomarkers, except in very restricted genetic subsets, end up really being subclinical manifestations of the disease once it's already developed. Yet we've known for decades that there are actually very early phenomena in the biology of coronary disease, and we've not really gone after those with any meaningful strategies. And so our goal was to move the entire spectrum of phenotypic analysis into the preclinical, or actually ideally into the wellness arena, so that we could begin to define at a cellular and molecular level the elements of transition to this disease. I think the benefits of doing this are we're focusing in an area where there hasn't been much work. Coronary disease has actually seen remarkable advances in the last couple of decades, with statins and now PCSK9 inhibitors having salutary effects on the disorder. Stents actually obviously having a remarkable effect even when there is an occlusion of an artery. But the goal would be to take an undifferentiated population, as you see in this right-hand panel, understand deeply how different forms of biology lead to the same final common pathway, build strategies that allow you then to identify these individuals before they develop disease. And in fact, I think that's one of the single most important things that we can do in biology, is to actually identify the people who don't have disease. For example, most of the studies of coronary disease, the control populations are contaminated with affected individuals who have just not yet presented at the rates of about 30%. We're really planning this as a generalizable strategy for phenotype discovery, identifying the known cell biology, moving it into the clinic, validating that in individual families, and then moving to populations or existing genotype cohorts, and ideally then understanding the core mechanisms of this biology. But at the center of what we're trying to do is really understand the perturbations that bring out this biology, whether nutritional or pharmacologic, that allow us then to democratize the phenotypes, as you heard earlier when Eric was talking. And the thing I think that's actually interesting here is understanding how to balance a portfolio. I talked about broad versus deep. I think we need to think about directed versus agnostic. We need to think about risk versus reward, but also think about how you maintain a cohort of investigators moving forward, despite the fact that not every one of them is going to be funded at that single time. To do this, we have a core infrastructure that is really based on taking ideas into the system, actualizing these, prototyping them, testing them, and then moving them into early clinics, both here at Stanford and at Brigham and Women's Hospital, before we begin to move them through discrete populations, and ultimately into that democratized culture. Could this work? Well, there are a couple of known phenotypes in coronary disease that we've identified decades ago, but not really built assays to look for. Cholesterol accumulates in the skin. There are abnormalities of statin response that are better predictors of mortality and morbidity than any of the known biomarkers. Why do we not just test statin response in a normal person to see if we can understand it? And then vascular abnormalities that have been present known to be present for decades that have never really been feasible to address. So the core impact, we believe, will be to identify fairly efficiently new pathways, new preventative strategies, using information that we believe we will leverage from new assays that we're adding to the system. Ideally, we want to learn generalizable lessons about how to expand this phenotypic space, which I believe, as you heard in the last discussion, is, I think, an important area of and framework for future advance. And the goal would be to think about a framework for a digital physical exam that represents the true underlying biology of the disorders that we're looking at. And this final slide really shows what I believe you heard in a couple of the other talks, that unless we integrate our discovery with care delivery, unless we begin to move the biology that we've known about into the clinic, we're really not going to be able to leverage the entire effort from the biomedical research and clinical and other communities. I pointed out in the last couple of slides ago that if you just plugged in the known cholesterol genes into a network from Drosophila from 30 years ago, you could have predicted that the genes would be the same as those for hypertension, diabetes, and cognitive decline. Why have we not moved that biology into the clinic? Thank you. This is the group of individuals that we're working with, not least Ewan and Stan Shaw. I'd obviously like to thank the leadership group, and Bob Harrington is the representative of in the audience today. Thank you for your attention.